Viral membrane. Human immunodeficiency virus, HIV, is a virus that attacks specific immune cells called T-cells. The anatomy of HIV plays an essential role in the replication and survival of the virus. The viral membrane is the outermost portion of HIV. This membrane is double-layered and comprised of fats called phospholipids. HIV glycoproteins. Specialized proteins called glycoproteins cover the viral membrane. These proteins enable HIV to attach to healthy cells, initiating viral replication. Glycoprotein 41, GP41, extends through the viral membrane, and glycoprotein 120, GP120, binds to GP41. Matrix proteins. Beneath the viral membrane is a layer of organized matrix proteins. Matrix proteins help the virus maintain its shape and protect the viral capsid underneath. Viral capsid. The HIV viral capsid acts as the HIV nucleus. The capsid houses HIV genetic information and the enzymes that help the virus replicate. Viral enzymes and HIV RNA. HIV is a retrovirus, which makes it capable of making DNA from RNA. Within the viral capsid is the HIV genetic material, RNA, and the associated HIV enzymes. These enzymes include reverse transcriptase, integrase, and protease. Reverse transcriptase converts HIV RNA into DNA, integrase incorporates HIV DNA into healthy cell DNA, and protease separates newly formed HIV proteins.